This is a production of Cornell University.
Thank you. Thanks for the introduction, and thank you all for inviting me to come and give your seminar. I'm really happy to be here and interact with more of you. I'm not a plant pathologist, but as Melanie said, a lot of the work that I've been doing is focused on plant pathogens, Pseudomonas syringae, in interactions with insects. So it's nice to be able to come over here and interact with all of you. So work in the lab and my interest in general is fairly broad. I'm interested in the evolution and ecology of bacteria interacting with hosts. Yeah, is the position under that's up here? In particular, I'm interested in how the bacterial genome content, gene content, connects to interactions with different hosts or in different environments. And in particular, this is interesting for bacteria that interact with multiple hosts or in multiple environments. So that might be in different abiotic environments, or bacteria that interact with a host and then also in other abiotic environments. And as many of you know, Pseudomonas syringae is a really good example of that because it does inhabit a lot of different environments. Of course, it's very well studied as a plant pathogen for good reason. It's a very common plant pathogen that likely infects possibly every plant or nearly every plant species. It is also a serious agricultural pathogen that can cause serious agricultural losses, such as in bacterial speck of tomato or kiwi canker or different highly virulent strains that infect plants. But in addition to that aspect of the bacterial ecology, syringae, or what we call syringae, is a very diverse group of strains that a lot of which are not very closely related to each other and can inhabit a lot of different habitats. So it can be more of what we think of as an environmental bacteria. It can be found in leaf litter, in particular in the water cycle. So it can be present in lakes and rivers. And it actually rises up into the atmosphere via evaporation and transpiration. And it's thought to be a major nucleator in a biological nucleator in clouds, which forms precipitation and then falls back down. So it can be very common in those environments. Syringae can also be common on plants without causing disease, either because it's on a plant that's not a compatible host, or because it's simply epiphytic on the surfaces of plants. And we think it's in this context where epiphytic populations of bacteria are on plant surfaces that it might be interacting with plant-associated herbivorous insects. So a couple of years before I started my postdoc work on this, there was one paper showing that some syringae strains could actually infect, grow within, and then kill pea aphids. So kind of adding one additional habitat to this already kind of impressive list, that potentially syringae could be using aphids as an additional host. And a lot of my postdoc work was focused on these two questions, kind of defining what the effects of the bacteria on the insect hosts were, and also because of my interest in trying to connect genomes to bacterial ecology, to figure out what the genomic basis for these interactions actually is. And I'll be presenting mostly this work today and getting in a little bit to the direction that I'm going now as we're starting up work here in the lab. Just putting this in a broader context and understanding how these interactions with insects could actually be impacting bacterial populations. So most of the work that I've been doing has been focusing on hemipteran insects, such as aphids and whiteflies. So these are phloem-feeding insects, so they insert their kind of straw-like mouth parts into plants and feed on plant sap. They're also major agricultural pests, and because they can cause a lot of crop damage, there's a lot of interest in finding alternative control strategies, such as biological control or bioactive compounds from different sources that might be able to control them as pests. So that's partly why I've been focusing on hemipterans. I'll mostly talk about pea aphids today, but actually have found fairly similar effects across different hemipteran species, which is interesting. So this is showing survival of pea aphids and whiteflies that have been exposed to two different strains of Pseudomonas syringae. Pathovar syringae strain B728A, and Pathovar tomato strain BC3000. I'm mostly going to be talking about those strains today, so you can kind of keep this pattern in your mind a little bit. So the dashed lines are whiteflies, and the solid lines are aphids, and it's showing the proportion of individuals surviving over time. So these individuals have been exposed to the bacteria in artificial diet, and it's a fake plant cell that they can feed off on for up to a week without too much harm to the insect. And the treatments either were control with just buffer added, or bacterial treatments that had a bacterial suspension added to the diet. And as you can see, the aphids and the whiteflies have pretty similar decreases in survival with both of the strains. With B728A, you see a very virulent response where the insects, the survival goes down to almost zero within just a few days. For BC3000, survival is significantly decreased compared to the controls, but not at the same rate. It's a lot lower. And I found this to be true not just in pea aphids and whiteflies, but also in other hemipteran species and other aphid species. You see the similar highly virulent effect with highly virulent strains. And also I found fairly broad effects across other unrelated insects. So for instance, we did an experiment where we fed lady beetle larvae with either infected or uninfected aphids, and the lady beetle survival was decreased on infected aphids, and their rate of development was slowed. So it wasn't quite the same. It wasn't as virulent as it is for the hemipteran insects where they were all dying, but there was definitely an effect. We also found an effect in a Lepidopteran larva, so a chewing herbivore that was eating food that had natural numbers of bacteria in it, where the survival of the larva was decreased and development time was impaired. So it seems like there could be fairly broad effects of syringae across different insects, although that probably varies depending on what the insect is. The only insect that I actually haven't been able to find an effect for is fruit flies. Drosophila and melanogaster don't care if you feed tons of syringae to them. The syringae doesn't grow and the flies do just fine. But interestingly, all of these insects, of course, are things that would actually be encountering syringae on leaves. So the fact that you see some kind of significant effect is potentially meaningful. And in fact, it might be a fairly broad pattern across other plant-associated bacteria that we just haven't found yet. So there have actually been 4 plant pathogens that have been found to also be pea aphid pathogens. So these other 3 have very similar effects to what we see with Pseudomonas syringae, where they grow inside the insect and can be highly virulent. So it's possible that these plant-associated bacteria are actually common pathogens to insects, but we don't really have a handle on how frequent these interactions actually are. And this leads into this broader question of how environmental bacteria actually impact hemipteran insects. So mostly insects like aphids are studied for their intercellular symbionts, which we know are incredibly important for the insects. But they have fairly limited microbial communities other than that. So if you take an aphid, surface sterilize it, and then use culture-dependent or culture-independent means to see what kind of bacteria they have in their gut, you don't find very much. You find like a few taxa and a few cells, typically things that are plant-associated. And people tend to think that that is because they're feeding on phloem, there's just not very much bacteria there, and these, like, having a gut community isn't something that's important for the insects. There have also been few known bacterial pathogens found, other than the ones that I showed you. And it's not entirely clear if that is because this is an actual pattern, that these environmental bacteria aren't interacting with insects that much, or if it's maybe an observation bias. So when the aphids get infected by syringae, they don't have any obvious symptoms. So there's a lot of fungal pathogens of aphids, and you can see when aphids get infected with fungal pathogens. The fungus grows out of the aphid, and it's really obvious. You don't see anything when they die from the bacteria. They just keel over and die. It's also potentially hard to find the infection in nature because the aphids are dying so quickly. So you need to kind of catch, in order to sample an aphid that would be infected, you need to catch it before the bacteria grow too much and the aphid dies. So it may actually be hard to find if people haven't been specifically looking for it. So to kind of get a sense of how frequent these interactions might be, we've done some work in the lab in particular looking at infectivity of syringae aphids. So how many cells does an aphid need to pick up in order to become infected? This was an experiment done by Kelly Clark, who was an undergraduate researcher with me at the University of Arizona. And for this experiment, we used B728A, so we used a highly virulent strain so that we would be sure to get a strong effect. And we did the infection in artificial diet so that we could control exactly how many bacteria the aphids were being exposed to. And we exposed them to varying doses. We used second and third N-star aphids, so these are aphids that are still developing, they're not reproductive yet. So that means that they're not getting the added variable again reproducing and not playing with the results. So we infected them for 24 hours, and then we tracked them for an additional 5 days, put them on sterile diet, so now they've been infected and they're just on diet with sterile. And we tracked their survival. At the end, we tested to make sure that we knew exactly what the bacterial dose was, that we tested the diet that they've been feeding on. And we also took a proportion of some of the individuals and sampled them for infection to find out what proportion were actually infected. And this was replicated 3 times with 30 to 40 aphids in each treatment at each replicate. It's a lot of dead aphids. And it turns out that mortality is dose dependent, which is kind of what you would expect. Higher doses lead to higher mortality. This is the probability of survival and over time. And the likelihood of infection also increased with the bacterial dose. So higher doses, and this is the log of the bacterial dose, led to a higher proportion of infected individuals. And the higher infection likelihood led to decreased survival, significantly decreased survival. So that means that the mortality is actually correlated with the chance of the likelihood of infection. So rather than, it's not the number of cells that you're picking up that matters, but whether you become infected. If you become infected, you will probably die. And in fact, it seems that you can become infected with a very low number of cells. So here we have the proportion surviving again over the proportion infected, and each point is a different dose. And we calculated the number of cells that these aphids likely ingested based on how much they were feeding and what the dose was. So the blue line is the control survival. These are all doses that didn't significantly alter the survival. And the red dots are all doses that significantly decrease survival of the insects. So in these 2 cases, these 2 doses, the aphids were likely only ingesting 1 to 9 cells, but yet had significant decreased survival. And this is actually in keeping with what was found in another system in Dickia, where they used different methods to figure out what the minimum effective dose was, and they also found it to be about 10 cells. So if the aphids then get infected with even a small number of bacteria, those can grow and they die. And of course, this is definitely in keeping with the number of cells that you would likely find on a leaf, which kind of puts this in more of an ecological context, suggesting that this could be something that's actually happening. We've also done experiments on plants testing the same idea. So here, the plants themselves were sprayed with different titers of bacteria, and then the aphids were introduced onto the plants that became infected on the plant. And at the end of the time, we counted the survival of the aphids. At the end of the time, we quantified the number of bacteria that were on the leaves. So at when populations were at 10 to the 4 or 10 to the 6 colony forming units per leaflet, this is on fava plants, there was significantly decreased survival of the aphids. And so these are population numbers that you could actually find on leaves in nature. This is certainly this was done in a greenhouse, but it's certainly something that you could imagine happening in nature. And in fact, we see infection rates on plants of about 10 to 30% of aphids becoming infected, depending on the conditions. So it's somewhat a significant effect here, where a lot of the aphids are potentially getting infected, suggesting that this could be common in nature. And it raises the question of, if there's these highly virulent pathogens out there that they're encountering frequently, what are the aphids doing to defend themselves against this infection? We haven't done anything with aphid immunity. Aphids are thought to have a reduced immunity compared to a lot of other insects. So they have a lower number of immune function genes in their genomes, and For a lot of pathogens, they don't really mount as much of an immune system compared to what we expect to find based on other systems like Drosophila. So it's kind of thought that aphids maybe don't respond immunologically. It's thought that maybe they have more varied non-immunological defenses, and so we've mostly focused on that. And one possibility is symbiote-mediated immunity. So intracellular symbionts can be defensive against pathogens. They can help the insect survive a pathogen infection, and we found some evidence for that actually in whiteflies. But another possibility that we were interested in is fecundity compensation. So fecundity compensation is when there's a reproductive response to an infection. And in theory, individuals can invest in reproduction over investing in immunity, and that might be favored when a pathogen is highly virulent. So if you're not going to survive an infection, there might be no point in trying to fight it off. And maybe you should invest the last of your resources in just reproducing as much as you can before you die. Particularly if you are an organism that can reproduce a lot. So aphids are asexual and have really high reproductive ability, so maybe it makes sense for them to be able to invest in defense in that way. So to test this, we set up an experiment similar to the one with B728A that we described before, that I described before. Except this time we used DC3000 because it's a lower virulence strain, so it doesn't kill the insects as quickly, so we could actually follow them long enough to watch them reproduce. So again, the insects were infected in artificial diet using different titers of bacteria, except after they were infected, we put them on plants so they could kind of be happy as an infected aphid can be. And we tracked their survival, development time, and reproductive rate over the course of a week. I'm just going to give you the answer to what happens to survival and development time. For survival, it was dose dependent, as we saw with the other strains. So with higher doses, more aphids died. There was no effect on development time. They reached adulthood equally fast if they were infected or if there was a lower dose. But we did see an interesting effect on reproductive rate. So this is showing the number of offspring per aphid per day. And the bacterial concentration, the log of the bacterial concentration on the x-axis. So each point is a different replicate, the mean of the replicate. And we had 3 replicates, and these are the control doses for all of those. They're all kind of clustered up here. They're fairly similar in reproduction. The extremely high doses are over here. Mostly those aphids died before they got a chance to reproduce, so that's why those numbers are so low. And with the remaining points, you see a significant positive correlation. So there's actually a decrease in reproductive rate with very low doses, and then an increase in reproductive rate with high doses. And these rates are actually higher than the control rates. So this suggests that maybe there is some kind of cost to infection that might be lowering their reproductive ability here, but possibly they're then increasing that reproductive rate to counter the infection or the cost of the infection as they go up, as the dose goes up. And when you look at that, when you look at the means of the reproductive rate, and you kind of break the doses down into low, medium, high, you can see that a little bit better. None of these reproductive rates are actually significantly different from the control, but you can see the increase with the dose, and also the fact that the low dose actually has a significantly different reproductive rate than the high dose. There's definitely something different happening with this low dose than with the high dose. And that's particularly striking when you look at the survival of the aphids at the same time. So this is the percent number of aphids that are surviving on day 7. So that's the point when all of the aphids have started reproducing. So at these high doses, there's less than 20% aphids actually surviving, but they're really reproducing at a rate that's higher than the controls. We think that the mechanism here might be that those aphids that were exposed to the high dose, so the black circles, are reproducing faster than the other aphids. So here there's a peak of reproduction actually at day 6 after infection, and then that starts to go down, whereas the control aphids, the open circles, are just kind of starting to ramp up reproduction. So they might be kind of increasing the timing of that reproduction to counter the infection once it starts to get bad. So this is some suggestion that aphids may actually be reacting to virulent infection with fecundity compensation. We also have been exploring another potential defense, which would be avoidance. So if there are these strains out there that are highly virulent, it would make sense that the aphids would, or insects in general, would evolve to avoid them. So this was work that was actually done by an REU student in the lab here, Rachel Fay, an REU student through the Microbial Friends and Foe programs in the summer. We're really excited about how well this worked out. Rachel was able to do 3 different experiments looking at how syringae influenced aphid feeding. So in the first experiment, she looked at feeding rates on artificial diet with or without different bacterial strains or control, and no choice. So in this case, the aphids were in a setup where all they had access to was a feeding sachet, and it either was a control or had bacterial suspensions in it, and they either had a choice to feed or not feed. And she followed them for 24 hours, checking every few hours to see how many were feeding. In a separate experiment, she looked at feeding preference. So in this setup, the aphids were given 3 different choices. It was actually in a 96-well plate, so each well could be a different choice, a different bacterial strain or control. And the aphids were free to roam around and go wherever they wanted. And she looked again at how many aphids were feeding over time. And of course, what we're most interested in is whether this actually works on plants. So we also did an experiment where they were given a choice of feeding, they were on plants, and they were given a choice of feeding on leaves that had bacteria or didn't have bacteria. And this is being done on fava plants, which are actually a really nice setup for that, because as the leaves kind of unfurl, each leaf is composed of 2 leaflets that are about the same size. So we painted one leaflet with epiphytic bacteria, and the other we painted with a control, and let those dry. And then we introduced aphids onto the plant and let them make their choice, and we counted them over the course of several days. So we did find an effect of B728A on aphid feeding. So this is the proportion of aphids in each cup that are feeding, either on control diet with B728A, or diet with BC3000. The control and BC3000 were the same, and feeding, but B728A was significantly less. And you can see that in the control in BC, pretty much all of the aphids are feeding at any given time, whereas with B728A, only about 30 to 40% would be feeding at any given observation point. And this holds up when you actually give them a choice between different treatments as well. So in here, there was less feeding on BC3000 compared to the control, although it wasn't significantly different. And there was almost no feeding at all on areas that had B728A. You might notice that there's definitely a lower rate of feeding overall here compared to what I just showed you, where almost all of the aphids are feeding. Here, only, I think, about 40% were feeding at any given time. This might be a difference just in the experimental setup. These were in a different container that was bigger. It might also be because this had more spaces that didn't actually have food, and so they had to move around and search for places that did have food. So they may have just had less time that they were able to spend feeding. There might actually be an effect of them being able to experience both the control and the bacteria. Potentially, if they found a spot initially that had bacteria in it and then they didn't want to feed there, they might move on and try to find other places and just, again, spend more time moving around, less time feeding. At the end of this experiment, we also wanted to confirm that these results looked right by testing to see if they became affected from that feeding that they were doing. We found a low rate of infection by BC3000, which matches what we're seeing. And actually, Rachel didn't find any aphids that were infected with B728A. I should say that our ability to detect a very low infection, if there were just like a few cells in the aphid, is pretty limited, because we grind it up and plate it out. And if there were just a couple of cells, we could miss those. But I think it's striking in that it matches these results really well, and it also suggests that maybe the aphids are actually actively avoiding the virulent strain. Because it's not clear to me necessarily if they might become infected with B728A really quickly and then stop feeding because they're infected and kind of unhealthy, or if they're actually, they encounter B728A and then they try to avoid that food altogether. And then I'm going to show the results of our plant experiment. This is kind of preliminary. We've only done this once, and so I'm not not saying that I believe it necessarily at this point, but it is in keeping with what we have found in that there were at least twice as many aphids feeding on leaves without bacteria compared to leaves with bacteria. And this is over the course of several days, so these numbers are fairly consistent. Which makes sense, the aphids don't really move around very much once they settle, but it seems that they were making some kind of choice initially. And there's a significant difference here. So I like these results. They're suggesting, in keeping with this idea, that there are broad and potentially very virulent effects on the insects. The fact that Hemipteran insects die very quickly from this infection, and you find effects on other insects as well. And also we're finding that there might be effects on more complicated insect behaviors, such as reproduction or feeding behavior, and things that we care about because also those affect plants. And I'd like to be able to, I'm going to continue exploring these ideas a bit more and getting a better sense on what exactly those effects are. But I'm also interested in being able to figure out what the genomic basis of these interactions is. And so this question has been looked at in Syringae for aphid or for insect virulence in a couple of different ways. In the initial work, that paper initially came out, Stavronidis et al. Had done assays with transposon mutants, so they had screened a number of transposon mutants to look for attenuated virulence. And they weren't able to find something that was a virulence factor that seemed to have a significant effect on virulence. But they did find that knocking out swarming motility in Syringae impacted virulence. So it might be important in some way. We've also tried some targeted knocking out of genes that look like they might be insect toxins. So Syringae has homologs of genes that are known insect toxins from Photorhabdus, and knocking those out has no effect. We also, I just started kind of screening all of the mutants that I could get my hands on that were mostly mutants for regulators of plant virulence, and found no effect there either. And as you'll see in a second, there doesn't seem to be a relationship between virulence in plants and virulence in the insects, so it's something different happening in those two cases. So this, because these approaches weren't really working, I tried a different way of getting at this. In this case, using the fact that there are a large number of sequenced Syringae genomes and a lot of phenotypic variation between strains, to do a genome-wide association study. So the idea here is that you would have strains with sequenced genomes that vary in virulence, some that have high virulence and some that have low virulence. Take all of their protein coding sequences and cluster those into COGs, into clusters of autologous groups. And then look to see what COGs are present in each strain and how that correlates with their virulence. So you would do a correlation between the strain phenotype, in this case virulence, and the abundance of genes in a COG that each strain has. So I'm going to present the results of this work today. It's somewhat preliminary in that I only have 12 strains that I've included, but it's kind of a proof of concept that this is something that you can do, and hopefully I'll be able to expand on more strains and get a better idea of what's actually happening here. In order to screen the strains, we screened 14 strains that had sequenced genomes and actually screened them in whiteflies for virulence. So these strains are a pretty broad swath across the Syringae phylogeny, including, I think, some strains that are no longer classified as Syringae. So there's definitely a lot of phylogenetic breadth here. And I'm going to show you virulence data for all of these strains just so you can get a sense of how that varies across strains, but I actually only included strains in the analysis for which I had 3 replica assays for virulence, so I can have a very good idea of exactly how virulent the strains are. And as you can see, it varies quite a bit. Some of the most virulent strains are, of course, B728A, which we talked about before, and actually 2 strains that aren't plant pathogens, TLP2 and 6-7, so that's why I think that virulence of insects isn't necessarily related to virulence of plants. This group of strains in general tend to be more virulent, whereas a lot of other strains can be less virulent. I should say that virulence here, I'm measuring it as the time that it takes for the bacteria to significantly decrease survival compared to controls. So pretty much all of the strains will infect and then eventually kill the aphids. And what kind of differs is how quickly that happens. So this is just one measure of virulence. You could do it in different ways, the total number that are dead at different times or whatever. And I've tried looking at virulence in measuring it in different ways. That way you get kind of the same results regardless. So I just like this method because you can more easily compare across strains. So taking this data, taking all of the strains for which I had good virulence data for, I clustered the protein sequences using OrthoMCL and then did a correlation of the number of genes in each cog with the virulence of a given strain. And so the idea here is to identify candidate genes that are correlated with virulence, either because they're present in highly virulent strains and absent in low virulent strains, or they might be more copies in high virulent strains than there are in low virulent strains. And what you get, this is some example patterns that you see for some of the most highly correlated cogs. If this is the number of copies of each of a cog across the strain going down in the columns. So you see cases where highly virulent strains like CYP7 might have multiple copies of a gene, and a lot of cases where the low virulent strains, of course, have no copies. And when you actually look to see what those genes are, the list of candidate genes, unfortunately, a large number of them are hypothetical proteins of unknown function, so I can't really say very much about that. There was one regulatory gene, a transcription regulator that might be interesting if that's changing transcription across strains. There was a chemotaxis gene, a signal transduction protein, that I was interested in because it seems that motility potentially is important for the virulence. And then a couple of other genes that we know their function, but it's not entirely clear how that might be related to virulence. And in fact, these are genes that are correlated with virulence in this analysis, but that doesn't mean that they're actually causing the virulence. There is likely, I haven't controlled for the phylogenetic signal here, so these strains are more closely related and, of course, more virulent and also tend to have similar genes in that way, so there might be an effect there that certainly needs to be considered. And of course, some of these genes can be present in these virulent taxa for other reasons, not related to the insect at all. For instance, a lot of the strains that are highly virulent, especially these that are in group 2, are more commonly epiphytes. They do a little bit better, it's thought that they do a little bit better in the epiphytic habitat. That is intriguing because it means that they're more likely to encounter the insects. But it also might just be a coincidence that whatever makes you a good epiphyte may also make you virulent to insects. And I'd like to be able to get at that question of, is this just a coincidence, or are these strains actually adapted for being virulent to insects? And this gets at my larger question of understanding how these insect interactions impact surrounding populations, and asking if the bacteria actually benefit from the insects at all. Are they getting something out of using insects as a host? Or is this actually a dead end for them, especially for strains that are highly virulent and they're killing their host within a few days? Is that something that benefits them at all? And theoretically, it could. We know that the bacteria grow to really high numbers inside the insects. So this is data from whiteflies. So these whiteflies were infected for 24 hours and then taken off the diet and allowed the bacteria grew inside the insects up to fairly high numbers for both strains at a similar rate. You get actually get higher numbers in cave aphids. Cave aphids are a little bit bigger than whiteflies, I guess, so you get more growth in there. Up to 10 to the 6 to 10 to the 8 cells per aphid, which is kind of huge given the size of an aphid. And we know that those bacteria can be excreted in aphid honeydew. Honeydew is kind of a picturesque word of saying aphid waste. So hemipteran insects are feeding on plant sap, and they excrete all that extra sugar in this kind of really sugar-rich liquid. And infected aphids can secrete a large number of bacteria in their honeydew. So Stavronidis et al. Found that infected aphids could put up to 10 to the 7 CFU per square centimeter of bacteria on a leaf. And he hypothesized that that could be important for the bacteria in terms of building up their population sizes, but also dispersing to other hosts. It's not clear to me if that's necessarily the case, because it might be good initially to be in honeydew because you're in this like sugar source, but as that honeydew starts to dry out on the leaf and the osmolarity changes, you're then in potentially a really bad environment if you're a bacteria. So to look at this, we were interested in looking at syringae persistence in honeydew. So we took infected aphids, we collected their honeydew over the course of 24 hours, and at that time, we quantified the number of bacteria that were excreted in the honeydew for some samples. And for the other samples, we took them and we incubated them at different humidities for an additional 24 hours and then quantified the bacteria again. So this is showing the change in bacterial population and CFU per aphid after that incubation compared to what the initial numbers were in honeydew as it was. So it's just persistence in honeydew without any additional input from the aphids. And the full change is on a log scale because there's a lot of variation. 0 would be no change, so the numbers are about the same as they initially were, the population stayed the same. And -1 would be all the bacteria died. So you can see that at 50 and 75% humidity, that bacteria don't tend to do very well in the honeydew, actually. And it's not until you get up to 99% humidity that you actually see some growth. So at very high humidities, potentially honeydew might be a beneficial environment. There may also be differences between the strains. B728A had like a higher mean than B3000, although there's a lot of overlap here, and I don't know if that would hold up when we have more data for this amount. But again, this is kind of reinforcing this question of whether interacting with insects is actually beneficial for syringae. Are they going to do better if there are aphids on a plant and there's honeydew being laid down, or is that actually a bad environment? And to look at this, we set up an experiment where we sprayed epiphytic populations of B728A on plants, and then we introduced aphids onto the plants or no aphids. We let the aphids be on the plant for a week, and at the end we sampled epiphytic bacteria on the plants and quantified bacterial populations. So this is just the results of one experiment, so it's still a bit preliminary. But as you can see, the bacterial numbers were higher at all humidities when aphids were present. At the very high humidity, it was actually significantly different, and at 50% and 75% humidity, there was overlap in the ranges. There's a fair amount of variation in the population size from leaf to leaf or plant to plant. But actually the means are quite different. This is a log scale, so there's a high amount of difference there. It's not clear to me if this difference is necessarily because the bacteria were growing in the aphids and therefore their numbers were increasing, or if maybe they are deriving some benefit from the honeydew on the leaves. What I showed you before indicated that they were only really getting any benefit at the high humidity, so I don't know that that honeydew on the leaves is necessarily helping them that much at these lower humidities, but that might be. We're going to be redoing this experiment with more samples to see if that holds up, or if we can get a better sense of how different these actually are when they're aphids present or not aphids present. So to answer that question, do the bacteria actually benefit from having the insects present? The answer is sometimes, depending upon the environmental conditions or possibly on the specific strains. And we're going to keep working on using experiments to really understand what those conditions are, what the environmental abiotic conditions are, or how different strains react to this interaction, and how the insects are actually impacting the bacteria. And altogether, kind of looking at both sides of this, both the insect and the bacterial side, we're getting a sense that there can be very complex dynamics of how syringae interacts, not only with plants, which of course we know, but also with other members of the ecological community, with insects. We have direct impacts on the survival of aphids, indirect impacts on the survival of predators to aphids. We know that syringae can apparently impact aphid feeding and aphid reproduction, which of course would have an influence on plants as it changes the aphid populations. And then over time, we're hoping to get a better sense of how the aphid also impacts the bacterial population, to kind of understand what those effects are and when they're meaningful. And then ideally, I'd like to be able to connect what we learn about these ecological interactions to the underlying genomes, not just looking at insect virulence, but hoping to find the genomic basis for other traits such as aphid avoidance, like what are the aphids actually picking up on in the bacteria that they're avoiding, if they are avoiding them, or what bacterial traits are necessary for success either in growth inside the aphid or for success on habitats when aphids are present. So those are some of the directions that the lab is going. I just want to briefly thank some people at my previous postdoc labs at the University of Arizona and Berkeley, and then at the new lab here, Kevin Gessler, my lab tech, did some of these experiments, and Chris Rachel Kay was our undergrad, and funding from IFTEC.
This has been a production of Cornell University, on the web at cornell.edu.